In the past, we've talked about different ways that variation is introduced into populations. So we talked about crossing over and independent assortment during meiosis and random fertilization. Now we're talking about a fourth way that variation is introduced into a population, and that's through, as you can see here, mutations. Mutations allow for variation in populations. Some are beneficial and some are detrimental. We're going to talk about that today. So first, let's talk about what is a mutation. A mutation is simply a change in the genetic information. It can happen in the DNA, it can happen in the RNA, it can happen in the amino acid sequence. So if you change the genetic information, if you change the genotype, you change the phenotype. And so here you can see there's a change in the DNA, and so we have a change in the appearance. Sometimes you can't see the changes, sometimes they're silent, and sometimes they're harmless, and sometimes they're harmful. In your DNA, this is pretty impressive. In your DNA, there are over 3 trillion base pairs on the human genome. There is 6 feet of DNA in each nucleus of each cell, and 100 trillion cells, about 100 trillion cells in our bodies. That's 113 billion miles of DNA. That can bring us to the sun and back 610 times. You are amazing. Don't forget it. You're an amazing machine. Mutations occur about 1 in 10 million. And so there are ways that your body works to fix these mutations. However, not all of them are fixed. If you watch the video that is right here that talks about the different ways that our body has evolved to fix the mutations, however, some of them are not fixed. And so that gives us about 320 mutations per cell that are not repaired. And so 1 in 10 million seems like a pretty good ratio. However, if you have 3 trillion base pairs, you end up having some mutations. Like I said, some of them are silent, some of them are beneficial, some of them are detrimental. So the ones that are problematic, they can cause diseases like cystic fibrosis, they can cause diseases like cancers or sickle cell anemia, which we'll look at in an activity later on. Some can be beneficial. If you look at the peppered moth right here, the peppered moth was originally white, and then during the Industrial Revolution, soot was deposited on trees, and so the trees turned black. There was a mutation in the moths where they started to turn black. This ended up being a beneficial mutation. The environment changed, and so the change in these species ended up being a good one because now the predators can't see the moth. And so the black moths are going to be more likely to survive to produce offspring. And so therefore, this is a beneficial mutation. Most mutations are subtle, and they result in things like baldness or wearing glasses. They don't give us superpowers like in the X-Men. So heritable mutations produce variations in the population. So just like we saw with the peppered moth, variation is beneficial. And just like we've talked about all year long. Mutations can occur naturally, but they also occur through mutagens. And mutagens are simply chemical or physical agents in the environment that cause mutations. So that would be like tobacco smoke, that could be UV rays, so please never smoke cigarettes or sit in tanning beds because your DNA is already going through many mutations without extra mutagens, so there's no reason to add more problems. You really want your DNA to be healthy. One type of mutation that we have out there is called a substitution. If this right here is the original genetic sequence, there was a mistake here and a G was substituted for the A. This is why we call it a substitution, because the G was substituted for the A. Now this can be no problem, or it could be a problem. Substitutions can be silent, and if we look at the codon CCC, if we look here at the codon wheel, CCC is our original codon sequence. Let's say that there is a mutation to the third base pair and it becomes CCA. CCA. Well, they both code for proline, so it's not going to be expressed. There's no problem here. This is called a silent mutation. However, sometimes the substitution can be expressed. So if we start off with CCC, we know that that's proline. If the substitution occurs at the first base pair, and let's say it's ACC, it results in a threonine. This is going to be a bigger deal than the silent mutation. Another type of mutation can be an insertion, and this results in something called frame shift. Make sure you write down frame shift somewhere in your notes because it is a key vocabulary term. An extra base is added to the DNA sequence. So let's look down here. If this is our original DNA sequence, or mRNA sequence, excuse me, we have the codon ACA, AAG. If there's a G that's inserted right here, we then have ACA, GAA. GAA codes for glutamine, or glutamic acid, I'm sorry. And then it changes everything after it. It codes for another glutamic acid and an arginine because all of the nucleotides got moved over. They were all shifted over, so this is called a frame shift. And this occurs when there's an insertion. This also occurs when there's a deletion. So if you look down here, ACA, we took out one of the A's, they got deleted by accident. Then we have AGA. All of the nucleotides got shifted the other way. So, and this is a frame shift again. And this is more detrimental than a substitution. It's problematic. So now this protein is sort of junk. And that is it for notes. I hope you have a great day.